Hey everyone! To become more familiar with how to describe polyploids, let's look at a few polyploid species. First is the banana, specifically the Cavendish banana, which is the variety that's most commonly sold and eaten. The genome is 3x equals 33. This 3x means it's triploid. The genome is AAA, meaning there are 3 copies of the A genome, which come from only one species. So the Cavendish banana is an autotriploid. One nice thing about this variety is that it doesn't have any seeds. If you slice it open, you'll only see a few aborted embryos, and the fruit is large as well. We'll get to why triploid bananas have large fruit and no seeds in the next few videos. So, if you've eaten a banana, chances are you've eaten a triploid. As is the case for many crop species, there are lots and lots of varieties of bananas. Some diploid, and some autotriploid as the Cavendish is. Some varieties are even allotriploid, with different varieties or species having different combinations of subgenomes, like AAB or ABB. You might not know this, but I spent a good decade and a half of my career working in research labs, and if there's one thing I learned during that time, it's that humans love coffee. Maybe a little too much. I'm a tea drinker myself, but I married a coffee drinker, so making coffee has definitely become part of my life. Coffee is also a $3 billion a year industry, providing millions of jobs in coffee-producing countries. Coffee beans are the seeds of the coffee plant. Most coffee comes from the species Coffea arabica. The beans are inside these berries, which are picked, dried, processed, roasted, ground, and brewed. Coffea arabica plants produce what people call arabica beans, generally considered the most desirable. Just for fun, if you're interested in the details of how coffee gets from plants to your cup or mug, I'll leave a link below this video for you to check out. Why am I talking so much about coffee in this video? Because Coffea arabica is an allotetraploid. Its ploidy is 2n equals 4x equals 44. The 4x tells us it's tetraploid, and the allo tells us that it contains chromosome sets from different species. Coffea arabica is the result of a hybridization between Coffea canephora, which produces the other type of bean used for coffee, Robusta, and Coffea eugenioides. I'm not that familiar with these species, so my pronunciation might not be perfect. Anyway, both these species are diploid, or 2n equals 2x equals 22. They're very closely related species, so they have basically the same chromosomes, numbered 1 through 11. Each species has 2 sets of these 11 chromosomes, for a total of 22 chromosomes. The karyogram for Coffea arabica looks like this. This tetraploid genome is comprised of 2 subgenomes, CC and EE, with the EE subgenome coming from eugenioides and the CC subgenome coming from canephora. In this example, canephora and eugenioides both had the same number of chromosomes, 22, and each contributed all 22 of them to arabica, which has 44. However, when an allopolyploid is formed, the 2 parent species that formed it don't always have the same number of chromosomes, and they don't always contribute equally to the allopolyploid. Consider the question, how many chromosomes could an allotetraploid have if the parent species are species A, which is 2x equals 8, and species B, which is 2x equals 12? Keep in mind that there are multiple ways a tetraploid can be formed. I'll give you a moment to pause the video and try to answer this question. Okay, let's first examine what we know. We know that an allotetraploid will have 4 sets of chromosomes, some sets from a gamete of species A, and some sets coming from a gamete of species B. The key here is realizing that there are multiple ways that this can happen. An allotetraploid is 4x and could be formed if a normal haploid gamete from species A with 1 set of chromosomes fuses with a triploid gamete of species B, which would have 3 sets of chromosomes, giving the allotetraploid offspring 4 sets. So, how many chromosomes could this tetraploid have? To determine that, we need to know how many chromosomes were present in each of these gametes. Species A has 2 sets of chromosomes. That's what the 2x means. If there are 8 chromosomes total and 2 sets, one set must have 4 chromosomes. So a normal gamete with 1 set would have 4 chromosomes. In species B, 2 sets add up to 12 chromosomes, so there are 6 chromosomes in 1 set for this species. A gamete with 3 sets, usually formed by the fusion of a 1x and 2x cell, would therefore have 6 times 3, or 18, chromosomes. Combining these gametes and adding their chromosomes, 18 plus 4 is 22. So one possible allotetraploid could have 22 chromosomes. The reverse could also occur, with a triploid gamete from species A fusing with a haploid gamete from species B. This combination of 12 chromosomes from species A and 6 from species B gives an allotetraploid with 18 chromosomes. Lastly, 2 diploid gametes, formed by non-disjunction, could fuse, producing an allotetraploid with 20 chromosomes. This last scenario is how Coffea arabica was formed and is the most common way allopolyploids are formed in nature. Note that all 3 of these resulting offspring are tetraploids. They all have 4 sets of chromosomes. But because they received different numbers of chromosome sets from species A and B, they have different numbers of chromosomes. Another example of an allopolyploid is wheat, which is an allohexaploid and is 2n equals 6x equals 42, and has the genome AABBDD. So when you're eating bread, most likely you're eating a hexaploid. Here is a karyogram of hexaploid wheat. There are many species of wheat, but this hexaploid species is the one that's most commonly grown for food. The A subgenome came from one 2x equals 14 species. The B subgenome came from another 2x equals 14 species, and the D subgenome came from a third 2x equals 14 species. Each subgenome has 2 chromosome sets, which comprise the 6 sets of the hexaploid. Another way to think about it is each diploid species that contributed its chromosomes to the hexaploid had 2 of each chromosome type, 2 chromosome 1s, 2 chromosome 2s, and so on. So that the hexaploid has 6 of each chromosome type, 6 chromosome 1s, 6 chromosome 2s, and so on. 6 of each chromosome type times 7 chromosome types, numbered 1 through 7, equals 42 total chromosomes. Hexaploid wheat was actually formed sequentially. An AABB tetraploid was formed first from these 2 species, and the D subgenome was obtained later to form the hexaploid. While we generally refer to organisms as being diploid, tetraploid, and so on, in some organisms, not all somatic cells have the same ploidy. This is known as endopolyploidy, which is basically when a diploid organism contains some cells that are polyploid. Endo means within, so endopolyploidy is the presence of polyploid cells within a diploid organism. Endopolyploidy is caused by endoreduplication, which is when a cell, in this case a diploid cell, replicates its chromosomes, but then does not undergo cell division. So the additional sets of chromosomes stay in one cell instead of being distributed into 2 cells, resulting in this example in a tetraploid cell. Endopolyploidy has been found in plants, insects, and animals, including humans. The human heart accumulates polyploid cells as a person ages. In fact, about 70% of heart myocyte cells are polyploid by adulthood. Think about that. There are polyploid cells working in your heart right now. Why are heart cells polyploid? Well, we don't know exactly, but one idea is that because replacement of heart cells by mitosis is relatively slow, at least in our species, the additional gene copies present in polyploid cells might make the polyploid cells more active, possibly compensating for cells that have been lost over time. Endopolyploidy seems to be particularly common in insects. One study of the giant ant examined the ploidy of all cells in various organs and tissues. They determined the percentage of cells that were diploid, tetraploid, octoploid, and so on. Take a second to look this graph over, focusing on this part of the abdomen. We see that only about 20% of cells in this part of the abdomen are diploid, about 30% are tetraploid, and about 30% are octoploid. So even though the giant ant is thought of as a diploid species, many of its cells are polyploid, going as high as 64 sets of chromosomes in a single cell. Okay, that's it for this second video on polyploidy. By now, you should be familiar with the various types of polyploidy and how the genomes of polyploids are described. As always, feel free to watch part or all of the video again, and in the next video, we'll cover how polyploids are produced. See you then.